be about tissue models of human corneas. Those are 3D tissues that consist of normal corneal epithelial cells, and they form like 4 or 5 layers of cells, and they parallel normal corneal tissues. We expose these tissues to 100 millijoule per centimeter squared from different wavelengths that were generated by a monochromatic source. So what I'm going to show you next is induction of DNA photodamage, and this was assessed 30 minutes after exposure. So we look at CPDs, and what you see here are the percentage of CPD in the total tissue, the whole tissue. So you see here the layers of tissues, what we can call like superficial, and then the wing layers, and then the basal. So what I'm illustrating here is the percentage of CPDs counted in the total. And then we did this experiment with or without tears. So as you can see, just by looking at the red curve and the red curve, it seems that in terms of CPD's formation, tears don't do much. However, let me point out that those are artificial tears. We used just water with actual PBS with some BSA, let's say a bunch of proteins. And when you look at how the penetration, so how these CPDs are distributed into the tissue, and then you look at the superficial layer, and then the wing layer, and the basal layer, with or without tears, maybe there might be some difference a little bit over here for this far UVC wavelength, but overall, these artificial tears, they don't really make a difference. They don't seem to affect the induction of DNA dimers. So we did the same for 6,4 pp's, and this is just a comparison, again, of CPD's formation through the tissue and 6,4 pp formation through the tissue. And you can see, even at for far UVC wavelength, you're gonna get some CPDs, but those are at the very superficial layer. So most likely these cells, these damaged cells, will go away. And you might be interested in what's happening to the basal layer, which, even for skin, but also for eyes, is where you might have very precious cells. And as you can see, 5 UVC wavelength, they did not induce DNA dimers at all in the basal layer. You might start getting some at 235 nanometer for CPDs, but a little bit at higher wavelength for 6,4 pp's. So 5 UVC wavelength did not induce DNA damage in the basal layer. Then you have to, I use these experiments to we did parallel experiments with previous experiments using models of human skin. So we wanted to expand those studies to models of human corneas. But at the same time, my quest is finding another biomarker, a more specific biomarker, that rather than just CPDs, to look at potential damaging effects. So we look at another type of DNA damage, gamma H2X foci, that are usually associated with double-strand breaks, actually with radiation, ionizing radiation-induced double-strand break. But I wanted to look at a different type of DNA damage. And as you can see, when you look at the whole tissue, so without going layer by layer, again, 5 UVC wavelength, they do not induce DNA damage, double-strand breaks. And then if you look at them layer by layer, again, you might have some in the superficial layer, particularly at relatively higher wavelength. So 24 hours after exposure, this far UVC wavelength will not induce persistent gamma H2X foci. So let me point out again, those are 24 hours after irradiation. However, in the basal layer, the highest percentage, about 10% of gamma H2X foci was induced by the 245 nanometer. Again, the parallel experiments in the human skin and human eye model this is what it looks like when you compare CPD's formation in the whole tissue, in the whole skin tissues, and in the whole cornea tissue. And then again, for the basal layer where we are mostly interested in, again, gamma far UVC wavelength, they do not induce DNA dimers even in the basal layer. With the caveat that, you know, the skin tissue has a nice layer of stratum corneum. The eye model doesn't, and it's actually, this is what happens in real life. So these data, just keep in mind that the structure is different. What I'm going to show you next is an analysis of other relevant biomarkers, again, 24 hours after exposure. So I was looking at cell proliferation. When the cell proliferates, it means that it's healthy, that it's good. So what I was measuring here, what you would see as those brown cells, they do express this protein, Ki67, and that means that they are healthy. And of course, you would expect just the cells in the bottom layer to be proliferating. So all the other cells, the suprabasal, appears to be differentiated, and they have exited the cell cycle, so they shouldn't express Ki67. So when you look at expression of proliferation, so you see that at far UVC wavelength, the proliferation of these germinative cells is really similar to control. You might have a little trouble with higher wavelength, again 100 after 24 hours after exposure to 100 millijoule per centimeter square. So far UVC wavelength did not halt proliferation of the germinative cells. And then you start getting some trouble with when exposure was from higher wavelength. And remember, these cells might already carry CPDs and 6,4-PBs. So it makes sense that might not be able to proliferate that much. And then in my quest for biomarkers, I was looking at cell death. And of course, my first idea was to look at apoptosis. And then I also look at some other markers of some other modes of cell death. That is autophagy or necroptosis. That is a caspase-independent form of cell death. And as you can see, 24 hours after exposure to 100 millijoule per centimeter square, far UVC wavelength did not really induce cell death. Cells are proliferating, they don't carry DNA damage, so they are proliferating. They don't die. The ones that die at higher wavelength, they preferentially die by autophagy and necroptosis, but not apoptosis, which I was very surprised. I was expecting also our previous speaker, of course, say that the cells in human eyes, they do preferentially die by apoptosis, by apoptosis. This is just what I was expecting to see. This is a control tissue, and I used an enzyme that chops the DNA, so mimics the apoptosis. So I really was hoping to find some of these black cells here to tell you that these cells were dying by apoptosis, but they did not. So we have to think about that. And I also look at cell senescence, beta-galactosidase, but I put it here to say that unsurprisingly, I didn't see many cell senescence. 24 hours after is too short. So we have to be aware of, in our looking for different biomarkers which could be more specific and with what biomarker makes sense, depending also on when you look at it. And then one final marker is the cell-to-cell communication, gap junction communication. What you see is in blue nuclei, and these tissues are they are all tilted. So the bottom of the tissue is up here and the top is here. Again, tilted, top and bottom. Again, top and bottom. So in blue, you see a nuclei, so it's where the cell is. And in green, you should see anti-connexin 43, which is a protein that is very important for cell-to-cell communication. And again, 24 hours after exposure to 100 milliliters per centimeter square, far UVC wavelength will not impair this gap junction cell-to-cell communication. Wavelength a little bit higher than 235 D. I don't have quantification here, but you just see like less green here, so you can, that's my statement. We will quantify that. So in conclusion, although these tissue models do not completely recapitulate a human tissue, they are very useful if you want to look at different endpoints and you want to look at different wavelengths as well. Artificial tears do not affect the induction of DNA dimers, and far UVC wavelength did not create damages for the endpoint that I was looking at. And I thank you all for listening and the team.